All right, we'll go ahead and get started. It is my absolute pleasure to be introducing Dr. Boe-hyun Kim today, who's going to be presenting. I've had the pleasure of working with Boe for a number of years on several projects that we've published together. Boe is an assistant professor at Mount Sinai in New York, where I think she's been since 2013 or 14. Starting as a postdoctoral fellow in David Volsky's lab. Bo's early work before coming to Mount Sinai really focused on prion disease, where her work was addressing the impact of prion both in cellular assays as well as in animal models. And then about 10 years ago, she moved into the HIV field. And her research direction really shifted to navigating the effect of HIV in neurodegeneration and neurocognitive disease. And as she's going to talk about today, she's further extended to study the effect of viral infection as a risk factor for Alzheimer's disease and in people aging with HIV. So I'm going to turn it over to Boe, who's going to talk to us today about EcoHIV infection facilitates AD progression in mouse models. So Boe, all yours.
Thank you for introducing me, and it's great pleasure to meet you all here today and to share my data, which I did with David together. So, can you see all the screen well?
It's on
slides yet.
Oh,
Oh,
there you
yes.
go. Yeah.
Okay.
Perfect.
So it looks okay, right? Yeah.
It's
So,
perfect.
Thank you. Okay, I will talk about today is what we are using. David's lab, we are actually using ecosystem with mouse model, and it's hard to use human model to study the mechanistic study. So I will talk about those models and how what we did with those mouse model today. So since everybody here is HIV people, so I will just go through quickly the first few slides with HIV stuff, because we know that HIV people population keep going up and growing every year. And the issue is after 1992, those people are not we are not having problem with the fatality issue anymore, but we have an issue with people are living more longer these days. And our concern is in our lab is because of their patients living more longer and they're getting more age are getting more older. So we are more concerned about this neurocognitive these days. We know we don't have any kind of very severe form of HIV anymore in the brain wise, but we still have a lot of problems with mild cognitive disorder and neurocognitive impairment issue wise. And we see there's like those hands people like 2.5% here, but eventually by aging when this population get aged, I think they're going to overlap with other field with dementia field together. So, and looking at the population growing with HIV patient is like, it's not only senior population, we keep seeing the new young population entering to the HIV field again. So this issue is going to be growing, I believe. So just giving a brief for what is, HIV-associated neurocognitive disorder is like, we don't see any kind of severe form anymore, but we see a highly prevalent brain disorder, and is one of the central health issue in people living with HIV. And they are chronically HIV infected individuals who can maintain low HIV burdens, and functionally they don't have any problem with and they're having lifespan has extended and tolerate to combination with ART treatment. And since the ART has reduced the death rate of HIV people and dementia, this patient has more like mild, they're entering into the more like mild format of HIV neurocognitive disorder patient group. Clinically, they don't have, they're not really clinically as a range of neurocognitive impairment. and they have a more mild criteria than HAD, we think. And compared to HAD, the main HIV neurocognitive impairment is they have they're characteristically including very minimal gross brain progress, and then neuroinflammation and low HIV brain burden, and then extensive synaptic dendritic injury without significant neuronal loss, and extensive dysregulation of neuronal gene expression. And as we already know about these patients are hard to study with living patients, and also have a limited number of archived brain tissues in our brain bank. And so our laboratory has developed an approach to study HIV and CI in mouse models. And we made a chimeric virus we can infect the virus and infect the mouse and study the mouse model for mechanistic wise. So what is the difference between HIV-1 and EcoHIV system is genetic wise, they're very intact, but we replace the GP120 envelope protein to GP80 so they can infect the mouse system. So biology of this ecosystem is this chimeric virus expresses all HIV genes except for GP120, which is HIV's envelope protein. And the GP80 can make this chimeric murine virus to infect mice. And echo-infected mice are seroconvert and mount protective CD8 T cells infection and then limiting viral replication systemically in the brain also. And they can be transmissible by mating, and infection of mice with ecosystem is preventable with clinically prescribed ART, and both intracranially and intraperitoneally inoculation of EcoHIV allows for efficient CNS infection and accompanying hand-like pathology, like infection of microglia cells in vitro and in vivo system. So the entire aim of our study is to investigate potential crosstalk between the process of aging and factors involved in prodromal AD and HIV-mediated mild cognitive impairment in people aging with HIV. So the focus of our studies are people aging with HIV are more prone to develop Alzheimer's disease. How we designed the experiment and what is our approach to the study is we use 3 different administrative approach. One is intranasal to deliver oligomer A beta to CNS, and intraperitoneal IP injection for systemic infection of virus, and intracranial injection to enhance the infection impact and shorten for developing the phenotype. We use 2 different type of behavior approach, and one is radial arm automase, and it looks like this way. And then we use fear conditioning system for assess the behavior and memory. And first part, we use the black mice to see the synergetic effect between oligomer A beta input and EcoHIV systemic infection. The design for this model, this study, we use black mice, regular black 6 mice with EcoHIV system by IP injection. And the timeline was we infect them on day 0 and we wait for 10 days for infection, and start the behavior on 10 days, but we give them 2 days before oligomer A beta through intranasal, day 8 and day 9 twice, and then we started behavior to see the synergy effect on day 10. And the reason we choose the day 10 was when we do the behavior kinetics by infection day with 1 day after infection, 10 day, 25, and 55 days after infection of ECHO system by IP, we can see that until that day 10 after infection, they're pretty close to the control system, and it's kind of like subclinical stage for the ecosystem. And after 25 days, it's shifting completely and they change the behavior. So we choose the day 10 to see the synergy effect of oligomer A beta and a systemic infection of EcoHIV together. And we do the titration of oligomer A beta to see the what dose concentration of oligomer A beta will have caused the synergy effect with systemic infection. So we choose, actually, we only see shown here a 5 microgram and 3.2 microgram of oligomer A beta here, but we actually titrated from a more higher concentration in the beginning. And here you see it's a 3.2 microgram. Even if we give them intranasally with infection, they don't change any behavior here. So we don't see any synergy effect by behavior wise. When we give them 5 microgram intranasal per mice, then we can see they're having synergy effect and start to change the behavior. So we decide to go using this 10 days after infection with 5 microgram of oligomer A beta intranasal system. So when we first time, we want, after giving 10 days infection, we wanted to see if they're affecting to their long-term memory synergy by long-term by measuring long-term potential. If they are, we can see the change in the memory dysfunction. So here we give, after 10 days infection, we take out the hippocampus and run the LTP and giving 50 nanomole of oligomer A beta. And when measure the LTP here, we can see this group is 10 days after infection with oligomer A beta given the hippocampus, and we can see their synergic effect and they're dropping the memory function here. So we can see they are dysfunction, their memory function is scrambled here. And we can see the,
yes? Excuse me, your cursor is not well visible. So it would help if you could point out to things, if possible.
You mean my pointer?
Yeah.
I don't know how
but
to
if
improve.
it doesn't work, it doesn't work. So just refer to, A, B, and C, and so
okay,
forth.
okay. So 10 days in in panel A, you can see 10 days after infection with oligomer A beta, and when we measure the LTP, then we can see their synergy effect with dysfunctioning memory effect. And we did the same things for the 30 days after infection, and it's still having scrambling the LTP here, but is there differences? What we can see that their start memory dysfunction with the oligomer A beta is starting very early time point here. And so next, what we see in here, so we're going to study this in vivo system in mouse model. So after we give an intranasal OA beta, we collect the CS from each mice and measure if we are actually delivering the oligomer A beta to the brain system. So we collect the CSF and measure them by the timeline, and then we can see from they start to picking up from like 30 minutes later and become high peak and 2 hours later, and it continues until 4 hours later. So we can continue to measure them. So we can see, actually confirm that we are delivering the oligomer A beta to the brain system. And here, and it's the behavior we did, and then it's the system with 10 days after infection with 5 microgram of oligomer A beta, and we have a 4 group here. PBS alone group, and PBS with OA beta group, and PBS echo with vehicle, and echo with infection with oligomer A beta intranasal 5 microgram. And we can see only those oligomer with infected group is having memory dysfunction. And when we go to the visible test for physical tests, and those all 4 groups has a no problem for vision wise, and they don't have any motor problem. So we can see that those systemic infection is subclinical stage, and oligomer A beta subclinical, those are having synergistic impact to change the memory dysfunction. And to check the viral load in these mice, we check the brain and spleen and macrophage together, and we can see the brain virology with oligomer A beta is actually elevating a little bit more higher than echo alone group. And macrophage also showing the same wise impact. And spleen, somehow we don't see so much great difference between echo alone group and oligomer A beta group. We tested what is actually causing this impact in the brain. So we wanted to see if they're affecting to the cell death. We did a ton of assay for these brain samples. We can see the PBS group have no apoptotic cell death shown. And PBS group with oligomer A beta 5 microgram alone, we can see a little bit here and there. And echo alone group, we can see the small portion of cell death in cortex area, but not in hippocampus area. And when we put together with EcoHIV plus A beta, then we can see there's a more massive cell death, positive cell death in cortex and hippocampus together. So when we actually quantify the cell death cells per same area, then we can see that those 2 factors are having synergy effect compared to alone groups in cortex and also hippocampus wise. And when we wanted to check in the protein levels for apoptotic cell death levels, so we choose cleaved caspase 3 in Western blot. So comparing those PBS groups with echo-infected groups, we can see their cleaved caspase 3 is actually going up more in oligomer A beta with infected groups, and they have a more synergy impact compared to the rest of the groups. So we think they're actually what they're inducing the memory dysfunction here is because of their causing more cell death, inducing cell death, that's the cause of the inducing memory dysfunction here. We checked this not just protein wise, we wanted to check this in gene transcriptome wise, and then we choose a couple of genes based on related to brain functional wise, and then we choose the PAN marker and M1M2 marker and trophic markers and disease association microglia markers. And then PAN marker, we checked IBA1 to see how much their glial microglial cells is activated in this way. And we can see they're actually increasing with echo infection together, not oligomer alone, but they're increasing, and the echo with OA beta together, they're actually elevating a little bit more higher. And for M1M2 is like pro-inflammatory and anti-inflammatory markers. We can see pro-inflammatory markers going up together with all the echo groups, and anti-inflammatory groups, we can see oligomer A beta alone group and echo alone group. They have a similar amount, but echo combined with OA beta together, then their anti-inflammatory markers are going down very synergistically. And trophic marker, we choose TNF alpha and NF kappa B and interferon gamma and IL-1 beta, and they're all going up in combined groups together, except for NF kappa B group. And we can see there's NF kappa B and IL-1 beta have a synergistic effect. And microglia related, we had a couple of CST7, CAT7, and ThyroBP, C3 and C1q and TREM2 markers. And when we check all these markers, actually we can see between these genes we picked, those anti-inflammatory markers and interferon gamma, IL-1 beta, and C1q have a synergistic effect with echo infection and oligomer A beta intranasal together. And this is until now we used our black mice, which is immunocompetent mice, and then we don't see so much. We don't actually found any plaque assay wise. So we want to see if we can see the phenotypes using another type of mice. So we choose APP transgenic mice, one of the familiar fat mice, actually familiar type of AD mice, and then we choose APPS1 mice. There's a one Swedish mutation in APP gene and preselenium1 mutated gene. And those mice have a certain kind of phenotypes in the plaque and tangles and synaptic losses and neuronal loss, gliosis and LTP-wise and cognitive impairment. What I can point out to for watching next slide is their plaques are start to deposit in 6 weeks in cortex in 3-4 months age, and their tangles are not mature completely, so we cannot see the tangles in these mice. And the other point is their neuronal loss is found at 17 months old animals in these mice, and you can find the LTP change after 4.5 months of the age. So we choose 4 months to termination timeline. So we don't actually in normal PBS, these APP mice, they're not supposed to change. We're not going to see. We expect not to see the LTP change or no plaque, something like that. So this is the reason we choose these mice. And those mice are 10 days infected by IP, and then we did LTP and fear conditioning, and we check other protein assays. And when you see this LTP changes after 10 days infection in APP mice, you can see that their LTP is changing dramatically. It means actually echo infection is actually accelerating the AD process here. It could be one factor to accelerate the AD process. And the fear conditioning behavior-wise, you can see the infected APP mice are actually, because it's 10 days infected group, and probably if infected alone group is not going to change the behavior with wild type. And you can see APP mice are having more, showing the memory dysfunction, so their freezing time is slower than other 3 groups. So we checked with this using these brain samples, we analyzed the total amyloid A beta brain protein amount and oligomer A beta amount. And we see that total amount of A beta protein is not really changing, but oligomer forms of the amyloid beta is going up more significantly in APP mice with echo infection group. So we can point out that one of the reasons they're changing the behavior and all the memory dysfunction is because of their oligomer A beta is accumulating pretty much a lot in the brain inside. And when we check the brain biology and peripheral biology itself, then actually the wild type and transgenic mice brain biology and spleen biology was not really changing so much in here. So I think the major issue was echo infection was causing to accumulate a lot amount of oligomer A beta in the brain, and it caused the memory dysfunction. And this is how we did the amyloid plaque assay using this APP mice. And this, you can see the left side on PBS group and right side is echo infected group brain. And they have like their termination time is 4 months old for 10 days infection by IP. You can see, so there, you can see some plaques in PBS group, but you don't see any inflammation around the plaques. And infection group, you can see plaques around. There's a pretty much a lot of inflammatory microglia surrounded around it. And when we check the plaque, when we did the plaque assay, we're counting this plaque, actually positive 6E10 positive cells, plaques, and then total number was not really changing. And when we divided the cortex and hippocampus, we actually, by 10 days IP infection, we don't see dramatically increasing significance in the plaque numbers wise. So neither, it could be because 10 days is not long enough to make, induce a lot of plaque formation, I believe. So that's maybe the reason that we don't see the numbers change. Quantity-wise, we don't see the big difference here. So until this part, I can say that HIV and amyloid beta synergy is inducing the cognitive decline more earlier time point than what we think. And EcoHIV infection increased neurodegeneration in AD-wise, I believe. And mechanistic insight into cognitive damage is this combining echo infection and OA beta impairs hippocampal long-term potential and potentiation and induce apoptosis in the brain without altering HIV level in the brain or spleen, which is highlighting a direct neurotoxic impact. So from this study, because we see no in plaque number wise, we don't see the great impact in IP infection with 10 days. We moved to IC system, injection system to shorten the time and to make a more great impact on the brain inflammatory issue wise. So we choose 8 weeks old mice and 3.5. We have a 2 time points here. So 2 weeks infection and 6 weeks infection by IC. And because we want to make it same timeline for termination time by 4 months old, we inject them 2 weeks. We create the 2 weeks infection on 3.5 months old mice. And for 6 weeks infection, we inject them when they're 2 months old and terminate them by 4 months old. The reason we decide 4 months old is because in APP PS1 mice, they start to accumulate this a plaque pretty much after 4 months later. And also LTP start to change 4.5 months later. So we thought to see our infection impact, and together within this mice, it's good time point to see the preclinical stage. It's good to choose the 4 months old here. So we start with 6 weeks and 2 weeks comparison, and we start with plaque assay here because you want to see if they're actually impacting to increased amyloid plaques in APP mice by time-wise. And actually 2 weeks infection, we don't see that dramatically difference in here as like a IP injection system. And when we watch the 6 weeks old infection, then we can start to see increase in cortex plaque numbers. And also we can see some of the hippocampal plaques are increasing. And we actually divided the group to female and male wise, and we can see the females and male also have a cortex has good impact to increase the plaques here. And this is how we actually enlarge the picture. Then you can see there are more plaques in infected groups, and it's a subclinical stage here. So APP mice 4 months, we actually don't see any plaques in PBS group so much. And you can see here that we see more hippocampus in the female. We have a more impact than male here. And to study, we need to look up the mechanism study. So to have a brief picture to why we are studying next level is because the starting point for starting A-beta forming the amyloid plaque is starting from the cutting the CTF99 format of amyloid APP protein, which is starting point for amyloidogenic process, and which is not formed by normal processing for APP process. So we thought looking at the CTF99 is a starting for the AD pathology starting point. So we started to look at it from this protein here. So this is 2 weeks infected mice by IC. And it has a mixed gender together. And in the infected group, we can see that they have a more significant amount of APP CTF99 proteins increasing in starting from the 2 weeks old infection. So we can see actually echo infection can accelerate the AD processing here. And to the time frame by 2 weeks and 6 weeks, we look at the oligomer A-beta accumulation in the brain samples. And we can see from 2 weeks, we cannot detect the protein levels in oligomer A-beta, but 6 weeks later, we can see their oligomer forms or O-A-beta start to accumulate in the brain. And we quantify them in total number wise and total amount and oligomer amount. And then we can see the oligomer amount is increasing significantly, and while the total amount is stay in the same level. And then we do more assays on 6 weeks of infection, because we can see the very big impact in 6 weeks infection. And this is actually, we've shown it before, but we just did the dot plot again. And 6010 is antibody for total amyloid beta, and this 811 for oligomer, and fibriform of A-beta, and this is skeptic for how skipping gene. And we can see, compared when we normalize total APP protein level with oligomer and fibriform, we can see the infected groups, their protein level is increasing pretty significantly. And this is this is oligomer form in 6 weeks. And when we check the intensity in this ploptane assay, we can see their oligomer form is going up significantly in infected groups. And we did an extra assay for the amyloid fibrous form with using OC antibodies. And we can see this infected groups are increasing the number of oligomers in here. So when they quantify the area percentage, then we see the infected group has more fiber forms than compared to PBS group. So actually we did the base scope to check if we can see a viral level in the brains. So we did the ice injection, the virus in the right side, and here actually striatum level. We keep track them for all the whole brains in 2 weeks and 6 weeks, and we can see the cerebral cortex 2 weeks, and lot amount in striatum in 2 weeks, and fiber tract actually here, the bridge between left and right, and hippocampus. And we can see from the 2 weeks a lot of mountains, certain mountains cortex, and a lot of mountains striatum, and some are in fiber tract. We can see the red dots are moving to this area. And 6 weeks later, we mostly see small mountain cortex and striatum still more, and then we see the lot more amount of the viral level in fiber tracts. Just based on this kind of phenotype, we can just guess that when we inject the virus in the right side here, then they start to move to the cortex and hippocampus from the right side, and using the fiber tract, they're moving to the left side and eventually moving to the left brain. And so we did analyze and quantify them by this checking the red dots and blue brown dots, which is IBA1, and red is HIV and brown dots are IBA1. And when we compare the virology, it is IC infected samples, but we just want to see if they're going to leaking out from the brain, out of the CNS to peritoneal. So we collect the spleen and peritoneal macrophage, and we can see that they're actually leaking out from the brain to the peritoneal. So we can see certain level of virology in macrophage and spleen. And because this is IC injected and we inject the virus in the right side, so it may be not very natural way, then it might be some people can think this is not the natural way that virus movement. So we actually analyzed the IP injected groups with virology, and when we inject them by IP and for 25 days and track them by dissecting the brains from striatum from right and left and hippocampus right and left, cortex right and left, and we can see actually there's a lot of movement in the right side of the brain, striatum, hippocampus, and cortex. And compared to DNA level, this left side is pretty much lower than left side. And RNA-wise, we can see striatum, and after 25 days, we can see the left side striatum RNA level either, but we don't see it in hippocampus in left and right. And the cortex area, we can detect only smell out in right side cortex. So based on this IP injected brain samples, we can see even if we do the IC injection artificially in right side, it's going to be the viral movement is going to be same as what we're seeing from our IC injected group and IP injected. So it's partially artificial, but their viral movement is going to be natural way to be. Since we are doing focus in AD-wise, so we want to see how the microglia are activated in CNS after infection. So when we talk about microglia activation, there's a couple of stages for their morphological change in microglia, and it just gives us picture of how they change the phenotype. This is how they look like in their resting state and hyperaminified, and they move to reactive shape, and then amoeboid and the rose shape. Is what is the stage is by sequence is by their activation rate stage. So in this picture is I just to show that APP mice 7 months old, this fat mice after 6 months later, they actually can form the large plaques with a lot of phagocytic shape of a microglia, which is like round shape here, like here, is pretty much amoeboid shapes. But because we terminate them by 4 months, we don't expect them to move all the way to here, I believe. I expect to see some of the hyper ramified shape of microglia. And we actually analyzed by IF staining and skeletonized those IF pictures. Then we analyzed their branch numbers and something that processing lengths and endpoints by cells. And then when we actually see the IBA, first we look at the entire whole IBA1 activated levels. We can see that both female and male wise, their percentage age of IBA1 plus cells are increased, and when we look at just count the same numbers, we don't see the difference in male brain, but we can see the significance in female brains. And when we make a skitolize and check the processing lengths and endpoints for each cells to analyze the microglia itself, then we can see their process length is growing and getting longer in both female and male, and endpoints are more growing, having many numbers compared to PBS to infected groups, both females in females, but not in males in wise. So we can think that infection is increasing entire microglia activation itself and their stage of the processing of reaction of the microglia in here. And we checked the gene levels here for neuroinflammatory markers wise. And we can see we did the similar genes what we did for black mice in the first part. So actually we can see their pan marker going up and homeostatic markers are staying similar and tropic markers are going up and in the infected groups, and most of this disease associated microglia are all increased in infected groups. So we can see that eco-infection is actually inducing inflammatory genes and also affected the activating microglia inflammatory here. And we did analyze the RNA sequencing analysis for inflammatory response. And in total gene, we can see that 245 genes are upregulated and 198 genes are downregulated. And this is hard to look at it, but I just want to point out from this, I'm going to magnify this E panel to explain more details about it. And we can see, look at it, this E panel, we can see a lot of NF-kappa B pathways and inflammatory response and ROS pathway and IL-6 and just the signaling pathway. And if we spread it out to each gene, then each gene is like signaling pathway. You can see IL-6 stressed and ROS and also inflammatory, and you can see NF-kappa B pathways are spreading genes here. And each genes I just signified market because which we are going to be very interested to look at it because those are some inflammatory genes and inflammasome genes, and which is we're going to be interested in the further study here. And to make a conclusion, what is actually finally make the memory change in this model, we evaluate the degenerating mature neurons by FJC staining. And compared to staining work with PBS and eco-infected group, we see that a significant amount of FJC signals are going up here in infected groups. And when we do this quantify by the positive cell per certain area, and we can see these infected groups are pretty much significantly increasing. So looking at this, I can say that our EcoHIV can activate C99 cleavage in the early stage in 2 weeks and induce OA beta accumulation in 6 weeks after infection. And microglia cell activation is induced in the early stage of HIV infection and induce neuroinflammatory response. And HIV infection can accelerate the progress of AD in prodromal APP-PS1 fed mice. And together, EcoHIV infection can induce early OA beta and fiber formation, which can then affect neuronal degeneration. Finally, these phenotypes contribute to accelerating AD progress in this model. And the conclusion with this black mice study and APP fed mice study, I will say that people living with HIV are now becoming people aging with HIV population, and HIV infected people can be prone to developing AD in the long term. And we should consider the joint contribution of aging and the long-term HIV infection, especially when the infection affects neurocognitive decline. So these works were a big collaboration between different groups, and I'm pretty much thankful for Dr. Volsky and also Dr. Potash because they lead all these big works and leading us. And we have a collaborator in Colombia, Dr. Aran Hughes' lab, and Elantina did a lot, and Hong did a lot of all these LTP works, and they did really a lot of work. And then on Mount Sinai, so internally, we have a Dr. Ehrlich's lab with Emily plaque assays and all this AD. She's a big person in the AD field, and we got help from Dr. Mugello's lab, Dr. Moray, she helped us to do all the part and sections in the beginning to do the assay stuff. And yeah, that's it. Thank you for attention.
Good. Excellent, excellent, Bo. Let's open it up to questions. I'm going to go to the chat first, and then we'll open it to others. Rama, I think you had the first question. You just want to unmute and ask?
Yeah, sure. Thank you, Barb. Bo, that was an excellent talk. Thank you. I have a naive, probably just trying to understand more. When you presented in the beginning about all the biomarkers that you were studying after giving an infection with EcoHIV, as well as the oligomeric A beta, I was trying to understand if the kind of markers or inflammatory cytokines that you're studying, are they very specific to this combination? Or would you, if you gave, for example, any other pathogen, coronavirus or anything that would also have a chance to get to the brain, would you see similar, or can you delineate between EcoHIV and other pathogens?
Okay. You actually have a question on this panel, right?
Yes, yes.
Okay, we choose it basically on what basically we can see more broadly from another different kind of infected disease too, but we choose this gene based on our RNA sequencing data. But we didn't see so we picked up those genes based on those sequencing data, but I cannot say these markers are only just for EcoHIV infected groups. And I have a thinking as what you are considering, that those genes can be more broadly impact in other kind of infection, as you say, is COVID infection, or can be affected these genes. And those genes or maybe this is not very only typical for or typically for only for HIV infection. But we never checked it with other kind of infection. So it has to be more studied in other field, I believe. Yes.
Okay. So general neuroinflammation is.
Yes, yes.
Okay. Thank you both.
All right, I'm going to try to go in order here. Arpan, I think you had the next question on the chat.
Yeah.
You want to unmute? Yeah.
Yeah, hi, great work. So I have a very naive question. So I try to understand means when you in first experiment, when you give the A beta oligomers intranasally and it accumulates in the brain, what is driving the increase in the HIV proviral DNA level in the brain with with the injection of the A beta oligomer? Means what probably may be the mechanism of increasing the proviral level in the DNA level in the brain. That's I try to, yeah.
I think. Oh, this one, right?
Yes, the F, yeah.
Yes. I actually, on that part, we are hard to address your question, but I think that when we deliver OA beta to the brain, actually they can affect the blood-brain barrier, and when they break down the BBB, then probably they allow more virus coming up from the peripheral, I believe, and that could be the case, that virology is going up in the brain. Because according to our ecosystem, their brain virology stabilized after 25 days. And this is happened when we 10 days infection. So they still have a lot of time window to bring the virus itself from the peripheral to brain. So I think that's the reason they're increasing more virology in the brain in this time point.
Okay.
We didn't check like 25 days worth after 25 days later, so I cannot say that time point, but probably what I can expect is 25 days later, probably there are not going to be big difference between just echo alone or A beta alone, probably. That's what I'm thinking.
Okay, sure. Thank you.
Sure.
Let's go to Ron and then Matt, who have their hands raised. Ron first.
Hey, thanks. I'm interested in trying to integrate this with the human literature. So currently, there's a couple of big epidemiological studies that have shown a reduced incidence of Alzheimer's disease, frank Alzheimer's disease in people with HIV. So if we were to try and compare what you've got here to what we see in the patient model, if you will, the things that are different are the substrate. So the host is different. The virus is a little bit different. Well, that depends on your perspective. But then also the big difference between this animal study and what happens in humans is antiretroviral therapy. So have you given antiretroviral therapy to these mice to attenuate the infection and see if you can attenuate the effects on amyloid deposition? And in particular, there's a paper that was published by a guy named Gerald Chun about reverse transcriptase inhibitors being effective in reducing the formation of amyloidogenic A beta or APP transcripts. So that's a mouthful, but the key is treatment with antiretrovirals, number one, and number two, specifically the NRTIs.
That one, we actually haven't tried specifically in this study, but we usually, when I work with ecosystem in this lab for a long time, usually in mouse model, ART prophylaxis works pretty perfectly to prevent the virology, but when we try to do the treatment, their effect itself is quite mild. I won't say there's no effect at all, but in viral inhibition wise, we don't see the big inhibitions when you try the ART after infection. So in treatment wise, we cannot, I don't think we can inhibit the biology itself by the ART in this model. So it's hard to address the question in this using this model, I believe. But since we've seen some of these genes are reversing by treatment, even if they're not inhibiting biology itself, maybe it's good way to assess another way to look it up for the future, that maybe there's another way to treat suppress the biology in the future, I believe. So we can consider to try it, I think, in the future.
All right, Matt, you want to?
Sure. Very nice talk. Two quick questions. The first is, have you treated these mice with anti-amyloid antibodies just to really confirm that in this model it's amyloid that's driving the learning and memory issues? And second of all, have you considered alternative pathways that might be at play? Like there's a lot of attention right now in the Alzheimer's field looking at lipid dysregulation, lipid dysfunction in the brain. And given that HIV is associated with lipid droplet formation, have you looked at any alternative pathways that may be contributing to the learning and memory challenges that you see in this model?
We haven't tried inhibiting using antibodies so far. The reason was related to addressing your second question, because our first aim was to create the model in the beginning. We were not sure if infection of ecosystem could be able to mimic the facilitating AD system. And we were not sure it could do it. And in the beginning, as you see in the first part, we were not able to create the plaque by infection itself in the black mice. So we were pretty disappointed in the beginning. And right now we are trying to use ecosystem to mimic, to have a replicate the phenotype of AD using different kind of mice right now. So right now it's hard to address that question. We haven't done it yet, so we are not able to address that question so far.
Good.
Well, we can consider to do it in the future. Yes, thank you for the comments.
All right, Bo, you got a couple more questions. I'm going to try to do this in order. We have two from the chat, and then Howie, I think will go next. So let's start with Philwa, and then Yusu.
Yep.
Yep.
Hello, Dr. Kim. Thank you for quite fascinating and transforming research. So I
Thank you.
recall, my main attention within our neurobiology, neuroscience, and neurology, that is actually still in synaptic plasticity. So I recall, actually, for example, oligomeric kind of, polymeric kind of existence within post-synapse. So that they're actually PSD 95, they're actually involving synaptic loss kind of mechanism. That's one attention within my interest. But actually overall, does your study do support or prove partially or in big picture for that still, in my understanding, posed in the scope of hypothesis, that amyloid beta hypothesis on the connection in causality to the outcome of Alzheimer.
Yeah, our goals for this study actually aiming to eventually to I don't know how David thinks, but what I wanted to do from the initiating this study was if echo infection can cause viral infection can be one of the cause to initiate the sporadic case of AD. So we wanted to focus in the beginning to focus on to create the AD-like phenotype by infection itself. And actually it's also, so we are more focused on right now to replicate the phenotype itself, especially people AD field and are focused on plaque. And I know there's an AD group, they have patient, they don't have plaque itself when actually they look at them. So even if they diagnose as AD, when they terminate it and they look at the pathology, but they don't have plaque, A beta plaque itself, but still the amyloid plaque itself is one of the best hallmark for the AD pathology. So we wanted to, in the beginning, when we started, we want to more focus and create the phenotype, and then to confirm we can do it or not. And that was why we don't have more, like, we didn't go into more synaptic plasticity stuff so far, but we do have a planning to do it because we do see the HIV infection itself can cause synaptic plasticity dysfunction. So we haven't gone we did some kind of start to study those synaptic plasticity, but we cannot clarify so far right now, but we do have a plan to do this in the future studies.
Yeah, sure. Thank you.
Thank you.
All right, we're a little after 1, but let's take the last 2 questions. Yusu and then Howie.
Yeah, thank you, Bo. It was excellent talk. I really enjoyed it. So I just have a very quick, maybe naive question. I would like to ask, like, when you prove that with the EcoHIV infection, like accelerate the A beta cleavage, and also have the microglia activation. And do you think that this cleavage is mediated by macrophage, or they're just not quite related? They're just parallel mechanisms. If it is from the macrophage, like microglia, then do you think like any pathogens or any conditions leads to the microglia activation can accelerate this process?
That's more delicate question, I believe, because it's specified the microglia cell types, right? And so far, we do like to study more microglia, and actually in the brain cell, the scavenging stuff, we we are more focusing in microglia, but I cannot say this C99 is cleaved by microglia itself, but those things we have to study more because those C99 itself is accumulating inside the neuronal cells, really, actually, not the glial cells itself. So in that part, I think we need to study more deeply to understand how we started but it's what we know for now is echo infection is initiating the cleavage for that fragment and accumulating inside and probably it's going to initiate the plaque accumulation, I believe. Yes.
And just to add, I would not exclude the possibility that HIV products themselves can be involved. That needs to be studied.
Mm-hmm.
No, Tato Neff.
Yes.
Howie?
Yeah, I have a comment and a question. First of all, thank you for a very nice and comprehensive talk. And it's always a pleasure to be working with and be in competition with David, because we've been together, David, for, I guess, close to 40 years. When I started working with you, I had a full head of hair and your hair was brown. So it's been a while. I'd like to go and answer Ron's question because I think that's very provocative. The literature is complicated vis-a-vis the role that Alzheimer's disease play in advanced HIV infection, especially in the face of antiretroviral drugs. Now, I might say to start, we have very different models. I'm sure you've read our papers. We're dealing with the generation of a mouse that has all the Alzheimer's gene, PS1, APP, and tau. We have now the triple knock-ins in the face of a NOG immunodeficient background. So we're dealing with more natural infection, but we get it. We get it, and we're studying it. I think the 3 points that I want to mention, and getting back both to your presentation and Ron's question, is number one, inflammation drives amyloid, both the monomers, the fibrils, and even the plaques. So other agents, other systems, certainly senescence and aging can affect amyloid, and we see plaques as early as the second and third decade. So the accumulation of this disease process occurs much more or before the clinical sequelae. And I think we all understand that. But the big issue is, what is antiretroviral drugs doing to this? And there's a fair amount of debate and discussion that certain classes of antiretroviral drugs can mitigate Alzheimer's disease. And we're studying that actually in our models and looking at the nucleosides, the non-nucleosides, and the integrase inhibitors and combinations thereof. And I think Ron is right on target. That was a great comment, and I think very important that the drugs themselves may affect the progression of cognitive impairment. The other issue, which is more of a question than a comment, is the humoral effects. There are drugs that actually, or antibodies that do affect certain forms and early the pathobiology of this disease, and that's something that we're all studying, really important. And not only the issues of the antibodies alone, but the combinatorial therapies, the delivery, looking at regulatory cells. Nobel Prize won 2 weeks ago on this, is how we halt not only the inflammation, but the oligomers, the fibrils, and the disease itself. I think we're going to be together on this for many years to come. Hopefully we'll survive time, because we're getting, at least David and I are getting a little bit older. But I think this is a very complicated phenomenon, complicated therapeutics, complicated pathogenics, and certainly complicated models in the face of progressive and combination antiretroviral drugs. So my only comment or question, and maybe I'm asking Ron or David about this, is that we got a long way to go. I believe. And this ain't going to be done in a two-dimensional manner. We're going to have to look at multiple facets of this process as the hand ages. I'll stop there. Thank you.
Oh, can I just respond quickly, Howard?
Of course, David.
I always enjoy your short questions, but another point I want to raise, that the average age of HIV patients on ART via, I think, World Health Organization data is 55 years old. That's very far away from being at risk of dementia. So I think Boe is right. As these people age, the risk will increase. So present results I think are not informative from this regard. We will, given that we survive, we'll see how it works in 20 years. Okay, so this is just raising this possibility. Thank you very much, Boe. Wonderful presentation. Thank you.
Thank you.
Yeah, the Boe, just to ditto that, I think is really comprehensive, organized, interesting talk based on all the questions you got. So thank you very much for presenting. Let me turn it over to Caroline quickly to talk about next week presentation and who's presenting. Caroline,
so
you
next
still on?
week,
Yeah.
yes, yep. So next week we have Kate Fitzgerald from Johns Hopkins, and her talk is OCT and OCTA in aging and HIV.
Excellent. All right. Well, thanks again, Boe, and thanks to everybody for sticking around late. Take all the questions. Thank you.
Thank you.